Good afternoon, everybody, and thank you for being here for our final speaker of this symposium. My name is Kwesi Apori, and I'm a PhD student here in bioengineering, working in the HER lab on clinical and point-of-care diagnostics. And it's my pleasure today to introduce our final speaker, Professor Samuel Sia from Columbia University. So Professor Sia is an assistant professor in biomedical engineering at Columbia. Professor Sia attained his BS in biochemistry from the University of Alberta in 1997, followed by his PhD in biophysics at Harvard University in 2002. And starting in 2002 until 2005, Professor Sia was a postdoc in the Whitesides lab at Harvard University, where he was also able to co-found a company, Diagnostics, which is a successful venture-backed company working on a microfluidic format for prostate-specific antigen detection. And since 2005, Professor Sia has been at Columbia as an assistant professor. So some of Professor Sia's awards include, obviously, the prestigious MIT Technology Review Innovators to Watch Under 35, as well as the NSF Career Award and the Walter H. Coulter Early Career Award. So the research motivations of the Sia Research Lab involve both 3D tissue engineering as well as point-of-care diagnostics. So within tissue engineering in the Sia Lab, they're studying the cell-cell communication as well as cell-environment interactions and precise control and design of the microenvironment, all in an attempt to rationally engineer and design tissue and organs from the ground up. And with respect to clinical diagnostics in the Sia Lab, the motivation is to develop low-cost and portable diagnostics for addressing global health in the developing world. So as somebody who's working in an academic lab, something I find very interesting is that often the technology development is motivated in very different directions. So trying to develop very novel technology versus trying to address a very pressing global healthcare problem versus trying to create a device for use in industry can often pull you in very different directions. And I think many of you will agree who are familiar with Professor Sia's work that he's been able to bridge these gaps quite successfully. In terms of technology development, his recent publications have been on lower power actuation and detection in microfluidic devices. And he's been able to test several of his diagnostics in the developing world on diseases such as HIV, syphilis, and herpes. And as I'm sure Professor Sia will talk about more, he's been involved with a very successful startup, Diagnostics, which has venture backing and I believe has some new products out that are quite exciting. So with that, I'll welcome our final speaker, Professor Sia.
Well, thanks, Kwesi. I thought you could have just kept continuing with the rest of the slides and probably did a great job for me. So thanks again, and thanks to Luke and Amy for inviting me here. So the work I want to talk about doesn't really need a lot of introduction. After the last three speakers, we want to miniaturize a lot of big machines, that type of testing, into something that you can hold in your hand. And so this is an example of what we want to do, a big machine like this, running some test into something that you can hold in your hand. In this case, this is actually a microfluidics machine. It's by caliper, but it has a big instrument around the chip into something that you can hold in your hand. And this is not our work. This is Anoop Singh's work at Sandia National Labs. But what we want to do is something similar to this kind of device. Now, as Kwesi mentioned, I actually didn't work on microfluidics until my postdoc in 2002 started with the Whitesides lab. And it was an interesting sort of time for me to enter the field because the advantage of knowing nothing is you don't really have any preconceptions about anything. So it's either fortunate or unfortunate, depending on how you look at it. I obviously knew about a lot of the great work that had already been done in microfluidics by academic researchers, including a lot of you sitting here today, as well as companies such as Caliper and Cepheid that were already started. But I went in thinking about this problem, which is why was there so many publications, so much work, even at that time, on point-of-care diagnostics, because even when the sort of the modern conception of microfluidics started around 1990, people already talked about point-of-care right around then, but the actual number of devices on the market for point-of-care was very small, pretty much zero, depending on your definition of microfluidics. And I was trying to understand what was the reasoning behind this disparity. And there were a couple reasons that I ended up concluding with, and this is something I talked about with Larry Kricka in this special issue on point-of-care testing and lab on a chip, where we were guest editors. But the two reasons I think have actually been brought up already, one by Paul this morning, and is that, first of all this instrument around the chip, they always like to show you this microfluidic chip with a coin beside it, but again, not all the stuff around it. And that's okay for a lot of things, because it's for research applications, you're running this in a lab, but it's not okay for point of care. And to say that at some point you can miniaturize that instrumentation into something like this sometimes just never gets there. And so I think that's one reason, the instrumentation. A lot of the approaches are just ultimately not miniaturizable in terms of all the instrumentation around it. The second point, I think, behind this disparity is actually something that John mentioned in the previous talk, is that all the procedures you have to miniaturize to get a functional integrated test at the end is not just one thing. So you can come up with a great sensing modality and then say at some point it could be a point of care test. But actually sensing is just one of maybe 10 or 15 things technically you need to do to get an integrated test. And so to put everything together in an integrated, coherent way, I think has been missing. And so those are two reasons, those are two sort of the pitfalls that we've wanted to avoid. And when I looked at this, my postdoc is something I thought about specifically for ELISA. That was the test that we originally wanted to miniaturize. It's a great test. But typically what you have to do is you have a big machine like this, liquid handling machine, and a plate reader. It takes hours to do, but it is the gold standard for a large class of protein markers in terms of the performance, the sensitivity, specificity, and so forth. But it's expensive. It's hundreds of thousands of dollars for these machines. It's not very small, and I don't think it's very fast. 3 hours or so is not very fast. Not simple to operate. Certainly you need to plug it in. And so we want to come up with something that could preserve the performance of ELISA, but has other advantages as well so that you can take it to the point of care. And so there are a lot of things that we had to do to get this test actually working. And so I'm actually just going to tell you about two of them that were published a while ago, and then I'm going to fast forward to where we are today. Originally, the work that one thing I want to talk about is the signal amplification. And at that time, we published this specifically for resource poor settings. That was what was on our minds, but and now, of course, everybody is also interested in this area. But the signal amplification for ELISA is not really compatible with continuous flow microfluidics. That is an enzyme conjugated to a secondary antibody, because when you're flowing and the enzyme is converting the substrate to the product, in a well, that's fine because the product just accumulates in a well. But when you're flowing in a microfluidics, all that product just flows away. And so it's not quite compatible with that. So we use this amplification chemistry, which has been used traditionally in immunohistochemistry, and Chad Merkin has used it for DNA and his product Nanosphere. But we thought it's actually a really great amplification chemistry to use for microfluidics for a couple of reasons. One is that when you keep flowing the solution through. So the way this works is we attach a gold nanoparticle instead of an enzyme to the secondary antibody, and then you come in with this silver reduction, silver solution plus a reducing agent, and then you get the silver film forming over the gold nanoparticles that's catalyzed by the gold nanoparticles. So it's good for us because number one, when you keep flowing the development reagent through the microchannel, the signal actually develops from the surface. So you just keep building. The more you flow, the surface just builds up from the surface. So this is compatible. The signal doesn't flow away. And number two, you are converting a signal that is 10 nanometer size. Typically with gold with nanoparticles, you need to use lenses and so forth to look at it. Here you're converting a signal that's nanometers to something you can see with your naked eye. That's actually millimeters to centimeters. So you're crossing orders of magnitude in size scale. And that's really nice. So here you can use AFM to look at the nanoparticles growing in size when the silver is reduced on top of it. So at some point, you actually just get a continuous film forming. And we just went down the street, started using these lenses and big microscopes and so forth. We went to Radio Shack and got a pair of laser diode and photodetector for about $5 and came back to the lab and measured the absorbance of the silver film. So with very, very simple optics enabled by this amplification chemistry, you can actually detect the concentration of the analyte, which is reflected by the thickness of your silver film and how much light actually gets transmitted. So very simple, and this is just a very early data at that point. So our assay, which was called Pocket at this point, basically performed in the same sensitivity and so forth versus benchtop ELISA as measured by 3 different signal amplification strategies by benchtop ELISA, except our test actually ran faster and the instrumentation was 1,000 times cheaper. Instead of hundreds of thousands of dollars in instrument, we got it down to about $100. And so you can actually see the result here with HIV serum. This is something, the silver, you can see a little bit of background, but if you have the analyte there, then you can see the silver film, millimeter size scale. You can actually just see it with your naked eye. So it's a nice signal amplification chemistry, but it's just one thing you have to solve. Another one is then how do you get all the fluids inside the chip? Because we don't want the person to pipette. We don't want to use a liquid handling robot. And so we came up with this technique, which is just really simple. It's almost embarrassingly simple, except it worked pretty well. And so what we did was we took all the reagents you need to run an ELISA assay, which included all the washing reagents and your detection reagent and so forth. We pre-pipetted them in the exact volumes and concentrations that you would run in an ELISA assay and separated them by air bubbles. And then we put all this stuff in a tube in this case. So what you do is you just plug the tube into your chip. This is a cross section. Plug the tube into the chip, pull vacuum, and the assay just runs itself, hands-free. And so you can deliver many different types of reagents for washing, for amplification. This is something lateral flow tests traditionally can't do. ELISA does, except we do this completely hands-free. So if you do the 6 channels in parallel, you can run an entire titration curve in 2 minutes completely hands-free. And so this sensitivity here is pretty low in 2 minutes, so you might want to boost it up with a longer time. But that was one idea behind this sort of reagent delivery. So a number of other problems had to be solved, and maybe a third one I'll mention is the manufacturability, because that's been sort of discussed. It's too complicated maybe with using traditional manufacturing technologies. We spent quite a bit of time trying to get this to work with injection molding. So we also use plastics, but we don't use laminates. We use injection molding. And the way this works is it's just 2 pieces of plastic that we bond together. Injection molding is a great technique for producing consumer products, very low cost, very high throughput, except if you want to put narrow channels inside injection molded plastic, it's not trivial. And so we actually spent quite a bit of time trying to get this to work, and we have it working now. And so we spun off a company called Claros Diagnostics, me and a couple of other guys in 2004, doing point-of-care ELISA, and it's based in the Boston area. And when we were raising money for this, because if you remember, we actually wanted to do this for the developing world. We were talking to investors back then, and we said, you know, this is what we want to do. And then they said, well, this is sounds like a pretty promising technology, and you know, this humanitarian mission is great, and I wish you the best of luck. See you later. So, you know, so then I was just thinking, well, that's not really what we want to hear, so let's keep talking. And so we eventually expanded the list of markers that we were willing to do. Of course, we knew that we can do this in the US and Europe market as well, but that's how we ended up working on this. Is among one of the investors we talked to was a group of urologists who looked at this and said, you know, it'd be great if you can do prostate cancer testing for us. And actually, I'll just maybe skip ahead here before I go back. This is the group of urologists that were working with the venture capital fund that eventually funded us. Really some of the top urologists in the world. Peter Scardino, for example, wrote the book on prostate cancer. It's really one of the best books out there. You can buy from Amazon. But they looked at us and they said, it'd be great if you can do PSA for a number of different reasons, and we agreed. And so we raised $12 million in venture capital funding since 2007, with the idea of developing prostate cancer monitoring as our first product and seeing whether or not we can do other products in the future. Prostate cancer is not really what I want to talk about, but it is the second leading cause of cancer deaths among US men. It's a huge, lucrative market, and I think going after a big market in the US is good because it really allows us to keep building this technology. And since then, we have built a manufacturing facility and clean room that's ISO certified, and this year we received regulatory approval in Europe. We got the CE mark. So I want to show you how it works here, because I have this instrument with me. And so it's right here, if I can just move this out of the way. Yeah, thanks. So this does have to be plugged in, because this is for the physicians, and they said they wanted something about this size. And I'm not going to do a live demo here because I actually ran out of functional chips, unfortunately. But you can see me online doing this in the last couple months. So if you want to see me hurt myself and prick my finger, you can do this either at MIT or NASA. I gave a live demos that I think that's archived. But the way this works, and I'll just maybe then describe it to you, is you simply prick your finger just like a diabetic patient, and then you simply load the blood into the sample holder, which has a tube inside, an anticoagulant. Just a few seconds would do the trick. And then you snap the sample holder into this plastic chip. And this only goes in one way, so you can't go wrong. So you just snap it in there. And this is the microfluidic card that we can produce millions of every 15 seconds down the assembly line for pennies in material cost per piece. So you can make these chips really low cost if you just don't do it the traditional way. And after you do that, then you can slide it into the instrument. So this has it ready, so just slide it in. And it'll recognize this as a PSA test. There's an RFID tag on it, and you press start. And the test takes typically 12 minutes. So what you see here is a countdown clock that tells you how many minutes are left. So this is counting down really quick because this is just a demo test. But usually this takes 12 minutes. And then at the end, it'll tell you that the test is complete, and they'll report a result back to you, 1.2 nanograms per mil. PSA is not really an easy test to do. I think it's actually a very good test case for us because you need to be very sensitive. We can do this down to 0.1 nanograms per mil. Not only that, we have to give you a number. We have to tell you the difference between 0.1 and 0.2. And so we can do that, and so we're pretty happy about that. And then you take this out, and you throw it away. So it's very simple for a patient to do. It's very simple for the physician to do. But to get this, even the part of the sample loading, if I was showing you pricking a finger and so forth, as I think John was mentioning before, it actually took a lot of engineering. So a lot of people say, oh yeah, eventually you'll prick a finger prick of blood, whole blood. This works with whole blood. It goes in and so forth. Getting that fluidics to not clog up and so forth took a lot of time, and I can't really take any credit for this because this was done at the company, led by amazing Vincent Linder, who was my fellow postdoc. But it's really great work, and it's approved in Europe, and so I think we're trying to do a couple things now, launching this product in Europe and also getting FDA approval in the US. So, but I want to talk about global health because that's what I'm here to talk about. So in 2000, maybe 10 years ago or so, I spent a month in a small village in Togo, and without any running water, without any electricity, and I was just sort of bumming around, actually. I mean, I was teaching English, but I wasn't actually doing a formal program. And so Tam and I actually talked about some of these internships. And so I know a lot of you are sitting here, a lot of students you've been to these developing countries. And for me, it was really a seminal moment for me because it made me think about what I wanted to do. And I thought, well, I wanted to maybe use my skills to not just go after basic questions in science, but also make a difference in the lives of the people in the developing world. And so then I did my postdoc, having that thought in mind, maybe not knowing exactly what I was going to do, but really inspired me to try to go down this route. So I really empathize with a lot of you who feel the same way, and hopefully you can find your own path also. But here, at the same time, just because the investors didn't want to fund this for Africa doesn't mean we can't do it. And so my lab at Columbia has been collaborating with Claros and a lot of other partners, which I'll show you in a minute, to make a parallel version of this test, which we call MChip. It stands for something, but it's not important. But we call it MChip, that works in Africa. The card is actually the same. It's right off the manufacturing line from Claros. So that way the company doesn't actually have to divert funds to work on this project. The investors are happy with that. They want something back for nothing, so that's what they're getting. They're gonna get a lot of great results without having to invest any money in this. So this is right off the manufacturing line. And I won't go through how it works. The filter paper there's to keep the blood and all the reagents in the chip at the end, so the reagents don't come out for biohazard reason. The instrument is actually quite a bit smaller than this one. It doesn't have a touch screen, so it's actually much cheaper. It's about $100 for that instrument, which has all the optics, all the electronics, and all the pumping you need to run this test. It runs on a single 9-volt battery. A single charge will run 50 hours of continuous usage. So it's an instrument that we have designed along with a couple of industrial design partners, Pratt Design Incubator and Smart Design. There was one of the panelists was talking about industrial design earlier this morning. We work with them. Smart Design actually makes these OXO products for your kitchen, among other things. They know how to make consumer products. And I paid, actually, an industrial design person to come with us to Africa and to survey people in the field to get a sense of what they want. And here's just some stuff that the industrial design partners came up with. I'm not really going to go through it, but it was the basis of our design that you just saw in the previous slide or here also. This is some of the guts of the instrument. Only point I want to make here is power consumption was something I looked at very carefully because I wanted really to work with a battery, and by that I mean a 9-volt battery, not a car battery. And so we looked at a lot of the power consumption, and we did little tricks in order to minimize the power consumption. For example, the pump actually takes up the most power, and for that, the way we solved that issue was to actually turn it on very briefly, but have a vacuum chamber inside to hold the vacuum. So we did things like that to minimize the power consumption. Now, we've been testing this in Africa for the last 4 years with a lot of people, but our main clinical collaborator is a group called ICAP, which is from the School of Public Health at Columbia. They have actually treated more more AIDS patients with antiretroviral drugs than any other group in Rwanda. They're very well known in the public health circles, but they deal more with treatment than diagnostics. So we've been piggybacking them off of them and their their treatment sites in Rwanda. These are 3 different sites that we've been working in Rwanda of various infrastructure levels. And I just want to show you a movie of our tests being run in the field. This is in Rwanda. So there's our instrument, and you open up the lever there and put in the chip, very similar to what I showed you with the prostate cancer machine. And then you would just close that lever, which engages the pump with the chip, forms a tight seal. And you press a button. So it's only got one button, it's only got one LCD display. It's hard to go wrong. So this is part of our design parameters. Here, 16 minutes later, you'll get multiple infectious disease results. So I don't know if you can read here on the LCD screen, but it simply tells you digitally whether or not you have the disease, positive or negative. There's no user interpretation needed like a lateral flow test. Multiple digital results reported to you in 15 minutes. And the further thing I want to show you, because there's a lot of interest in mobile health, is that communication module on the right, that box. Because that box actually contains a cell phone chip, which we've built, to communicate our results in real time, seconds later, to a central server. And if the cell phone network is down, it has a second backup system to communicate through satellites that are orbiting the Earth. So you can be anywhere in the world. You can send these results to a central server. In this case, it's done through satellite, so you get an email. And so you can be anywhere on Earth. You could run this test, seamlessly integrate it with patient records, and very low error due to data transcription also. So one of the issues with being in point of care is you could just lose your result, or you cannot integrate it with the patient records. Here, you don't have to SMS the results. Maybe you can SMS the wrong result. You simply hit a button, and the results will be sent. It's seamless. We think that's important. Not only that, but every result actually comes with a timestamp and is geotagged. So you know exactly when and where you did that test in case you forgot. And the data transmission is also very secure, either by cell phone or by satellites. And so we're working with a program called TrackNet in Rwanda, which is a PEPFAR-funded project, has tracked over 40,000 patients in Rwanda, AIDS patients. And we think adding STD data to that electronic medical record system might be interesting. So what infectious diseases are we testing? We did a field survey in various countries, and HIV, Hep C, and syphilis among the STDs we looked at for antenatal care came out fairly high. And so we've been working with HIV, hepatitis B, hepatitis C, syphilis, and herpes. For all of these diseases, the clinically accepted test is ELISA, so we're good to go in terms of the clinical validation. Finger prick of blood, load a button, press a button. 15 minutes later or 20 minutes later, you'll get multiple results. And per test is actually much less to make, but I think at the end it's going to cost about $2 in packaging. The instrument is $100 instead of, say, $100,000. And I don't really want to go through the results. These are some results that were done in a lab, not in the field, but they work pretty well for all three diseases. So our results have been submitted. They've been in review. I haven't published anything on this for the last 4 years on this particular project because we've just been working hard on it. We've submitted the first of these studies. It's now been reviewed for 6 months, but I don't want to talk about it yet because it's not out. But this is the gist of that initial set of studies. Multiple STD results in 15 minutes, no user interpretation needed. This is useful for maternal-child health, so-called pregnant mother-to-child transmission of HIV, syphilis, hepatitis B, C, and herpes. And the results that we've gotten so far really show that the results are as accurate as ELISA on hundreds of patients in Rwanda tested in the field. So you can bring a microfluidics product like this to the field and get it to work. And for these diseases, the drugs are actually available if diagnosed. It's not a drugs issue. For syphilis, the drug is one shot of penicillin. So we're not talking rocket science here. But the problem when you're in these villages is you don't know who to give the drugs to, and you can't send these patients back to a clinic. So if you diagnose them in the field, you can treat them. And not only that, but you can get real-time data synchronization with electronic medical records. And so I hope to be able to show you the actual data behind this pretty soon, but you just have to believe me for now on that. So one issue that comes up is the cost. I think we've been talking a little bit about that. So is this, okay, that's fine. You could bring this into the field, seems to work okay. Is it cost effective? So maybe a couple years ago, the Gates Foundation funded an independent third party, RTI and Commons Capital, it's an NGO and a social venture capital fund, to do an analysis of our technology, of this test, to see whether or not it would be cost effective and what is the public health impact in 3 countries, Rwanda, Tanzania, and India. They went to the field, interviewed people, looked at our price points, asked them, okay, is this going to work? Is this going to be cost effective? So the only thing I want to show you is just 2 things. One, deaths avoided and disability adjusted life years avoided. We can save millions of lives within 5 years given a certain set of assumptions around the world. Millions of lives with this existing test. And okay, that's fine, but is it cost effective? Well, if you look at the cost effectiveness, one way to measure this is the cost it takes to save 1 disability adjusted life year. That's a public health metric. That's a WHO metric they use to see whether it's cost effective. These are some of the most cost effective interventions known in public health. Immunization, oral rehydration therapy, and water and sanitation. It's about $1,000 or so, maybe a little less. The cost for DALI avoided for us is about $10. So, the catch is that you have to kind of get this project to a stage where you can actually sell it and so forth. But so when I hear cost and so forth, I think the reference is this possibly the cheapest thing out there possible? The reference for us has always been, is this something that's going to make a difference clinically? Because right now there's nothing that's being used. And if other approaches come later that are cheaper, that's fine. But we want to solve a clinical problem, not having academic exercise and comparing real and hypothetical technologies in terms of what, so that's something I think a lot of us academics are actually seeing, but especially those of us who are working in this translational field, but not most, such as some grant reviewers and so forth. So I'm not going to name names, but I hope it's going to change because I think, especially in engineering, what we do is not just academic. It's also to make a difference. But clinically, so these are some of the partners we've worked with, and I won't go through all of them, but they've all been really helpful for us. ICAP and Claros. are a couple. And so just to summarize this part of the talk, M-chip, same accuracy as ELISA, but much faster, minutes instead of hours. Finger prick or less blood instead of venipuncture. And this I would show if I had the real data to show. Well, I do have the real data, I'm just not showing you. But we did this down to 1 microliter of whole blood, which is something you can't even do with lateral flow tests. And this is important because, so for example, in babies, it's really hard to get 10 microliters. And I think Bernhard alluded to this as well earlier. You need to have things, have tests that use very low volumes in some cases. Cost of equipment, 3 orders of magnitude cheaper than ELISA and pretty cheap per assay. So I think it's pretty low cost. So let me just talk about a couple of other technologies in terms of device development. And then I'm gonna wrap up the device part of this. So that's the test that, even though I haven't published anything on it, is actually the furthest along and probably we've worked the hardest on. And so hopefully I can share more data with a lot of you in the future. I just want to show maybe 2 things that are a little bit earlier in the pipeline. One is CD4 counting. This is something that John has really done a lot of great work on and a lot of other people because it is the decision point at which you decide whether or not to give someone drugs, not whether they're HIV positive, but whether they have a low CD4 count. We've been funded by the Coulter Foundation for 4 years to do this, and we published a study on this using chemiluminescence, which I think is interesting because the optics there is simplified. You don't need any light source at all. And so potentially down the line, you can build a sort of a low-cost test for doing CD4 counting, but it is tough. So one thing I've learned from doing CD4 counting in the last 4 years is that it's pretty tough. So we're earlier than where John is and earlier than our immunoassay project, but we're trying to find some paths to go with this technology for cell counting. So that's just one thing I want to show you. Another one that I'm really excited about actually now that I do want to share with you is also published is valving. And so Paul and Bernhard talked about this earlier today with Micronics and their valving is if you can do valving, you can do a lot of things. And certainly with what Steve Quake and others have shown, if you have these multi-layer valves, you can do a lot of really cool things, probably everything you'd ever want for point-of-care diagnostics. The problem is that these valves are typically, number one, not easily manufacturable, and number two, the instrumentation around it is not small. And so for example, the Quake valves, you need a gas tank, and you don't want to be carrying a gas tank around with you. So that doesn't work. So this, we've at least looked at half the problem, which is the instrumentation. So instead of using, for those of you familiar with these 2-layer valves, there's a control layer, there's a flow layer. Instead of using air in the control layer to control these valves, we put in fluid. So it's actually a hydraulic valve instead of a pneumatic valve. And this concept actually has been proposed by Shu Takayama's group and published by his group. So we're trying to just build a little bit off of that as well. But the reason we do that is that now when you have the solenoid, if you look at part B there, right here, you can have a solenoid pressing down a membrane and it'll direct that force, the water, to this very fine layer. And by just the fact that this is a very small area and that's a very big area, you don't need to move a lot in order to actuate that valve. So this plunger you can also actuate very easily with battery power. So the whole system of a gas tank and computers and so forth we replaced by this instrument here, which is powered by a 9-volt battery. So here I have a coin, but it shows everything you need to operate these pneumatic valves. And so these are the exact same valves as the Quake valves. You get very precise on-off control. Using this kind of actuation technology, the response time is great. You apply the voltage, it will move right away. You can do multiple valves that are synchronized with each other as well, and so you can look at multiple ones and see that they work together. Very, very simple assay. It just shows that you have to mix 2 proper things together to get a signal in this case. If you don't mix them together, then you get a negative signal. Not the greatest assay on earth, but it does show that you have to do fluid handling properly in order to run an assay. And so the fact that we can run these multi-layer valves in the field, I think opens up a lot of interesting possibilities, and I'm very excited about that. In terms of manufacturability, because PDMS is maybe easier to manufacture than maybe a 23-layer sort of plastic, but it's still not the greatest thing on earth. We actually have some ideas on that as well, which Hopefully I can share with you sometime down the road. So I'm really excited about that, if you can actually get valving to work like the quake valves in the field. So in terms of the market, lab to market, the PSA test is actually on the market now in Europe, and other immunoassays are further behind. But for global health, I think we've basically finished what I call preclinical validation, but we still have to do a proper clinical trial. CD4 and the valving are a little bit earlier in the game. And this is a sort of a philosophical slide that I sort of added in response to some of the points that were brought up this morning. And so with the work that, for example, Paul talked about in terms of microfluidics 2.0, because I get asked this everywhere. I go, what do you think of the paper stuff? And so I always have to say something about it. So this is, I don't want this to be, this will probably get me in trouble, but let me show this. Microfluidics, I think, if it's 1.0 is what Paul referred to, then I would say microfluidics 0.0 is actually lateral flow. And so if this is sort of timeline 1.n and 2.0, planar, paper-based, then I think, paper-based microfluidics now is really an attempt to improve. It's not, I don't find it interesting. I do find it interesting. I really like the work, but I don't find it interesting because it takes place on paper, because that's actually been done it's actually the oldest thing that's been done. But what's interesting is that all this work now is trying to improve the performance of the lateral flow assays, keep its simplicity, but improve the performance. And what we're trying to do, I would say, is take a lot of the great work that's been done by just sort of like planar microfluidics, and we want to reduce the complexity and make it cheaper. So you can call that either 1.n, maybe I can call it 3.0, because we're trying to make this less complex versus make this perform better. And I think the way we're doing it is not exactly the same as other people are doing it either. And so that's sort of the way I think about it. But on a broader scale, I don't think there's any one winner or any one loser. There's going to be more than one approach that's going to be appropriate. So people love to say, oh, how are you better than this or whatever? Well, right now there's nothing in the field on diagnostics. So I'm not measuring what we do against any other technology as much as against the clinical importance. And it's not only this, but you have other interesting technologies like digital microfluidics, for those of you who are in microfluidics, like droplet-based stuff. And there's a lot of different approaches, and I think at the end, anybody who thinks that there's going to be one single approach to do everything is misguided. I really think so. So I think there's going to be a lot of interesting approaches, and there's no need to argue with each other. I'm calling for peace, I guess. So, but the issue I'm faced with, I think, is in our case, money for microfluidics, I believe, can save millions of lives. And this is not just a shameless plug for what we do, but I think for a lot of other technologies that are actually relatively close to the market. Not in 5 years or 10 years, there are microfluidic technologies close to the market. And we can do this cost effectively, as I've shown you. The instrument is 3 orders of magnitude lower. The per test cost by WHO criteria is very, very cost effective. So how can people look at it and say it's not cost effective just because there might be something more, something that is a little bit less developed, but that could be cheaper. So that's kind of my feeling. I compare this to a clinical standard, but the problem is that there's not a lot of money right now for clinical validation. And both at the NIH and the Gates Foundation, they keep they want to fund things that are new and what is the innovation? People always say, what is it? If you published this 5 years ago, it's no longer innovative, even though you've shown that it's a path to success. That's not really that innovative. In the first world case, you can get money for clinical validation through the private market because there's a return on investment. But right now, if the private market is not funding this, if NIH is this is not their thing, Gates is not funding it, believe me, I've talked to them, then who is funding it? So I think that is a problem for a lot of us that are going to, at this logjam, and we can keep filling the space with more interesting new ideas, but what about the ideas that have actually been shown to work? I'm not so sure how to do that. So that's part of my, just this first part. So how much time do I have left? Maybe 10 minutes, okay, perfect. So that's the end of the device stuff that I want to show you for now. So actually, I just have 2 more things I want to show you that talk a little bit less about device development than about maybe why things work the way they work in microfluidics and in diagnostics in global health. So number 1 is a lot of us who build these devices tend to just kind of do it, and then if it works, then we just say, okay, that's what worked. That's the justification. Why do we use this flow rate? Why don't we use another flow rate? I don't know, because it works. And we do modeling, and Paul and Bernhard with their H filter, they've actually been, I think, one of the few cases where they've shown both a computational and also a device. But a lot of other people just don't bother because they just want to build a device. And I don't really want to go through this because this is just too technical to go through at this being the last talk. It's a bit of a technical paper. But for those of you who are interested in this, I'll just refer you to the paper, this manuscript, which I will talk about briefly. But people ask, why microfluidics? Oh, because it's fast, because you consume small volumes, sample, it's sensitive. But the issue is in a real world setting, you have always finite sample and finite time. And so there's trade-offs. And so we looked at this and we said, well, under real world settings of finite sample and finite time, what is actually the advantage of microfluidics as it applies to, say, finding an ideal flow rate? Can you answer the question of what is the ideal flow rate in an assay? And so we did corresponding experiments, one by CFD simulations, and then the exact same experiment with the same flow rates and all these parameters that as best as we could in a simple fluorescence reporter system like this, and compared side by side the results. And so here's just a thought exercise. What is the best flow rate in microfluidics to use? Well, if you think about it, actually, these are actually CFD results. Well, if you have unlimited sample and unlimited time, it actually doesn't matter what it is because you get saturation under all conditions. So actually the answer is it doesn't matter. But if you have unlimited sample but fixed time, then you want to be a faster flow rate because here you're in a transport limited regime. So you can keep flowing, but you're always transport limited, you're never saturating the surface. You have to be above some flow rate. So for these boundary conditions, you need to be at a high flow rate. But here, if you have unlimited time and fixed sample volume, then it's a little bit different because if you flow things too fast, then you run out of sample and you actually never saturate the surface. So a simple question like that, like what is the ideal flow rate to use in a heterogeneous immunoassay, actually really is not a terribly simple thing to think about. Depends on your boundary conditions. So I don't really, again, want to go through this in detail, except to just give you the gist of this. One thing we looked at was also the physical regime, whether you're in the transport limited or the reaction limited case. And we found that, so not only are there these two regimes, but if you have these real world constraints, there's a third regime, which we call reagent limited case, because you actually run out of reagent if you have finite sample. And that's what's going to limit your signal. And so there's actually another sort of consideration you have to have. And this is, the reason we did this is because a lot of times you kind of want to be in a reaction limited regime. You never want to be in a transport limited regime in microfluidics, but is that really true? And so two quick sets of results. One is, let's look at the signal. Under what conditions will you saturate the signal? And remember, I showed you those three thought experiments. You don't really know the answer up front. Here, we found by CFD simulations that there's a curve like this, depending on how much capture antigen you have on a surface, but you still have the same shape of the curve. Turns out you will saturate the surface up to a certain flow rate, but after which you will start running out of sample in your assay before you can saturate the surface. So here, you actually want to be about at this point because you want to run the assay as fast as possible while saturating the signal. And experimentally, we found actually the same curve. And this was unexpected. We didn't know that it was going to turn out like this. But it was sort of interesting to sort of get that correspondence in the results. So that's sort of the answer to what flow rate. The other thing I want to point out is the physical regime. And here, we looked at the ratio of the bulk concentration to the surface concentration, which can tell you whether or not you're depleting the bulk concentration. Because if you are, then you're transport limited. If you're at 1, then you're reaction limited. And it turns out, under all conditions where you're saturating the surface, there are conditions where you can be significantly away from the reaction limited regime. So you can be transport limited for a huge part of the assay. As long as at the end you're reaction limited, you can still saturate the signal. So, so to sort of assume that you always want to be reaction limited is not actually always true. So here's just some lessons. We have a new regime. There's a critical flow rate in some cases. We also looked at issues like if you have a short concentrated plug versus a long dilute plug, but the same amount of analytes inside, what's a better way to do this in our assay? And it turns out the short concentrated plug generally gives you the higher signal. And you can spend considerable time actually in a transport limited regime, which actually could be to your advantage in some cases and still saturate the signal. So this is just taking a little bit of a step back and asking us why it works the way it works, and maybe that will help you engineer the device as well. And the last thing that I want to show you is on the flip side of this, I think there was a question this morning on markers, biomarkers, because all of us have been talking about devices. So I just want to talk about discovery of biomarkers for a second, because while biomarkers are available for a lot of diseases in the first world, for certain neglected diseases, the bottleneck is actually the lack of biomarkers. And a lot of people just don't bother looking for them because the market is so small. And so this is the process of for discovery of drug targets. So this is for drugs, not diagnostics. And traditionally, is a little bit ad hoc, but more recently, the way to discover what are good drug targets, including for neglected diseases, they use a more systematic approach where they use a sort of a funnel, high throughput screening. Let's look at the hits, hit to lead and so forth, and at the end, maybe you'll find a few that are validated. But do this in a systematic way and see what you get at the end. The problem is this approach is not really validated for any discovery of a drug target, but is something that a lot of people are working with. We want to do the same thing with diagnostic targets. And this idea was actually proposed to me by a couple people, Rosanna Peeling, who's now at the London School of Hygiene and Tropical Hygiene, and David Roos at UPenn. And so I've just been doing it, but it's sort of their inspiration. Because they've been working on this database funded by the WHO TDR called TDR Targets, which uses informatics through leveraging all these genetic genome sequences that are out there for pathogens and running through the genomes to see what genes are actually good drug targets. Instead of just doing this, which costs a lot of money, maybe you can use informatics to identify what are drug targets, because the characteristics of drug targets are actually somewhat known. Not so for diagnostics targets yet, but we want to see whether or not we can do something similar. And so that's one of the problems. That's our first problem, is what makes a good diagnostic target a good diagnostics target? So if you have a virus, why that particular antigen is a good diagnostics target and not another one? Usually, again, we just gloss over it. We're like, well, just because, I don't know. It's good, and so the clinical information says it's good, but what makes it good? So we want to identify those criteria. And then we want to back calculate, put the criteria into the genome databases and extract out other promising diagnostic targets. It's kind of like Amazon. If you like this book because of these criteria, you should also like these other books. And we think if you find this diagnostic target is actually useful, why not try looking at these other ones as well? So we looked at a whole series of molecular criteria, starting with tuberculosis. So molecular weight, hydrophobicity, glycosylation state, and so forth. Just go down the list using various databases that we think are reliable with the help of TDR targets. And we also curated the list of TB antigens that are known to be relatively good. And we saw what are the good properties about those promising TB antigens. I wouldn't say they're validated, but they're promising. So 3 things: subcellular localization. Good diagnostic targets should be secreted, right? Or they should be on the membrane. It's obvious. Well, actually, extracellular, for the ones that are sort of promising TB targets, are actually biased against an organism-wide baseline. So there is some truth to that, but look at all the ones that are cytoplasmic as well. So just because you're a cytoplasmic protein doesn't mean you're a bad diagnostic target. So some truth to it, but it's not absolute. Expression level, well, they should be highly expressed. That's obvious, right? Well, if you look at the various stages of TB infection and look at the expression level that people have characterized in databases, you'll see that in some cases they're highly expressed, in some cases they're not highly expressed. So it's a little bit more complicated than that, but there is something to be said. One thing that was a little bit unexpected was the isoelectric point. For the ones that are in red are the promising TB targets. They actually seem to have a lower isoelectric point than the baseline sort of organism-wide TB antigens. And so we're trying to figure that out. It might have something to do with the macrophages and so forth, how TB is actually taken in to the body. So we're working a little bit along those lines, and we hope to be publishing those results soon as well. So that's all I have to say about global health diagnostics. The other half of our lab works on tissue engineering using microfluidics, which I'm not going to say anything about, but we actually have published probably more work in that field in the last few years. So these are the people that have worked on the projects I've shown you. A lot of people, just the key people I want to show you at Claros, my lab, industrial design partners, clinical collaborators, and also TDR targets, the funding agencies. Thanks for your attention.
Hi, Dan Laser. Very nice work. I really enjoyed all the technical discussion as well, but I want to focus for a moment on the health economics slide that you showed, which was interesting to see, and those kind of analyses are not easy to put together, and I think they're pretty useful, especially in a global health context where policymakers are more likely to pay attention to them than, for example, a U.S. clinical laboratory director who has different financial incentives than the total cost of care. But anyway, the specific question I have was just to get a sense for why the result was so different between Rwanda and Tanzania, which obviously being geographically proximate and similar in GDP per capita, it seemed like I think the Tanzania number was 6 times that of Rwanda.
Yeah, so it's a good question. So that analysis, I have to say, was done by a third party. But the methodology was based on the set of studies that were published in the Nature supplement by the Gates Foundation and RAND, which some of you may have seen. And we used that methodology to do the analysis. But to your specific question, part of the parameters that affect the analysis is also the prevalence of the infectious diseases in the country. Because if there's very low prevalence, you have to diagnose a lot more people to actually make a difference. And so that's part of why the cost to save a certain disability-adjusted life year actually changes from country to country, because the prevalence is different in those countries. So, and maybe I'll just add one more thing to that is, so how is it possible that our test was so much more cost-effective? And I think if you look at the analysis, one reason I think is because the syphilis, for example, syphilis impact that we think we can make is actually quite substantial because it's very cheap to treat syphilis, very, very low cost. And so the problem really comes down to diagnosis. And you treat pregnant women who can lose the newborns due to congenital syphilis. So the result is you're saving an entire lifetime worth of lives for a very low cost. And so to rationalize why our test showed such high cost effectiveness, I think that's one reason also.
Thanks. I really enjoyed the talk as well. If someone came in and separated the M-chip wing of Claros completely from the rest of the company, could it survive and be sustainable and successful as a business that develops a product for the developing world?
Yeah, that's a great question, Tanner. So with that study that was done by Vita, funded by the Gates Foundation, we have now a 100-page business plan. We don't actually have any funding from the Gates Foundation or R&D, but we have funding to do an analysis, so we have another report. But the report says, so I didn't show you the financial statements actually, but as you would expect, the financials actually come back such that the answer is yes, we can be a profitable business, but the margins are going to be really significantly lower than, say, our prostate cancer test in the US or any other test in the US. So is it going to be hugely profitable? Probably not if you just stick to the developing world, but maybe this standalone business can try to sell the infectious disease product to the US as well and try to make money that way. But as a standalone business in the developing world, it's marginally profitable, which might be good enough.
Thank you for your talk. It was very interesting. And I was wondering if, in the development of your system, if the biomedical waste that's generated was a concern. I understand that a microliter of blood is very small, but is there a decon procedure that you attach with the instrumentation so that there aren't all these little packets of blood that just get thrown away? Because I think in developing countries, trash is a major problem, and it either ends up in the ocean or just dumped somewhere in a field. So have you guys addressed that at all, or is that not a concern?
That's a really, really good question. I wish you didn't ask it. Actually, it's so good. But it's actually an issue because with a lot of these devices, with say the plastic chips, the current way they're being disposed of is through incineration, which is not ideal. So I think it's one of those issues we have to face. Let's we save people's lives first and then see what happens. But we are actually trying to do something like that. So for example, Rosanna Peeling is very interested in looking at biodegradable plastics, and we might actually be all publishing something at some point on talking about that issue, looking ahead a little bit. So it is a concern. But an issue with using biodegradable plastic, if I can just jump maybe 2 steps ahead, for our system is that a lot of our features are really small. And a lot of the times when you use biodegradable plastic, they degrade over time, right? That's what happens, is that you lose the feature over time as well. And so it's a double-edged sword. So the answer is actually yes, we have thought a lot about it, but I don't really have a great solution to give it to you right now.
Thank you. I got to say, wonderful talk on a whole bunch of levels. Let's see if I can, is it actually on? Yeah, okay. It really is great stuff. And I love your slide where you sort of, 1 and 2.0 and 3.0 and 4.0 or whatever we're getting to. But I got to say, what you did, which we didn't do in my group, was to say, okay, it's got to be simple. It's got to be a low-cost manufacturable. And the problem, I would say Micronics, when they did their version of the nucleic acid amplification one, and even their version of immunoassay, which was a little less sensitive than one we developed, is they did start from the idea that it's got to be very few layers. The trap that we had is we worked with graduate students, and graduate students are really bright people, but what they want to do is get their degree. And they know that if Jaeger says, oh, it has to have this other function, what they do is they add another 3 layers. And they say, so that's how it got to be 23 layers. What I'll call microfluidics 1.n does not always inevitably get you to 23 layers, as I think you beautifully demonstrated. So I'm delighted about that. I got to say, though, people may ask you, what do you think of that paper stuff? What I get asked is, so how does what you're doing differ from Whiteside's doing? I even have a slide, all just to answer that question so I don't get angry.
Yeah. Everything comes back to George Whiteside, doesn't it? Yeah, in some ways. Thank you for the comment, Paul.
No other questions? Let's thank our speaker again.
I'm really delighted this first Berkeley version of the Global Health Symposium finished well. And I'd like to just thank first all the students from Amy's lab and my lab, and also staff from SAND. I really appreciate all their great help. Actually, it's from bottom up. Actually, it's Berkeley style. All the students mobilize, and actually, it is quite amazing that we didn't have any funding from neither Bill Gates or, or NIH for particularly this particular area, but it's coming along, and students really want to try, and they have more motivation. So I'm so thankful that all students from both our group participate well. And I'd like to really thanks to all speakers and also panelists. Actually, it's not too, thanks to last but not the least, I'd like to just thanks to Tamina. So if you come up, we'd like to give you some gift.
I don't know. Yeah, so I guess also I wanted to thank Tamina in particular and Sen, because they've, in addition to throwing this conference, which is great for all of you guys, they also funded my research in South Africa this summer, and they've also funded my research earlier on too. So all of that came through SEN. I really appreciate that.
Other research for sure.
Yeah, they fund a lot of stuff. So we got you this really, very stylish. It's what all the cool kids wear now.
Yes, thank you.
We got. Hold on, don't spill it.
Oh, thank you. To
Wow,
share. All right, I think we have a reception outside.
That's right. Yeah, so just to echo Luke's comments, thank you all for coming. There's a reception outside. Please continue on the conversation and dialogue. Thank you.
Thank you.